Alright, so today we are going to discuss the phases of mitosis. There are four main phases in mitosis: prophase, metaphase, anaphase, and telophase. So before mitosis, remember that in interphase there were three stages: G1, S, and G2. In the S phase, DNA was replicated to create an entire new set of DNA. In G2, the cell begins to manufacture the materials required for mitosis and cytokinesis to begin. By the end of interphase, the chromatin consists of two identical sets of DNA, which you will be able to see when our chromatin condenses. There will be two identical chromatids called sister chromatids, creating that famous replicated X chromosome shape, which we will see in prophase. So prophase is our first phase of mitosis. And during prophase, the chromatin condenses into tightly packed chromosomes, which you can now definitely see in those X shapes. The nuclear membrane breaks down and releases the chromosomes into the cytoplasm, and also the nucleolus disappears as well. One pair of organelles called centrioles, which are right here, they move apart to opposite poles of the cell. And as these centrioles move apart, a network of fibers, which are made up of microtubules, we call them spindle apparatus, or as you can see here, they're also called spindle fibers. And these spindle fibers form between the two centrioles. These spindle fibers lengthen with the addition of microtubule subunits. And this is how these spindle fibers get longer. Alright, so in our second phase we have metaphase. And during metaphase, the spindle fibers guide the chromosomes to the equator or the center line of the cell, as you can see here. The spindle fibers from opposite poles attach to the centromere of each chromosome. These spindle fibers attach in such a way that one sister chromatid faces one pole and the other sister chromatid faces the other pole. Alright, so now we move on to the third phase, which is called anaphase. And during this phase, the centromere splits apart due to the pulling of opposite directions from the spindle fibers. The sister chromatids then split apart due to this pulling of the spindle fibers. These spindle fibers shorten, and I want you to think about this. I've already stated how spindle fibers lengthen. Now, how do you think spindle fibers shorten? We will address this question in tomorrow's class. So as these spindle fibers shorten, the sister chromatids are pulled to opposite sides of the poles. And at the same time, other microtubules lengthen and force the poles away from one another, as you can see in the center line of the cell here. Alright, so in our final phase of mitosis, we have telophase. Telophase begins when the chromatids have reached the opposite poles of the cell. The chromatids begin to unwind into longer forms of chromatin, and they become less distinctly visible. The spindle fibers break down, and the nuclear membrane forms around each new set of chromosomes. And also a nucleolus forms within each nucleus. You can't really see that in the diagram here, though. We can also clearly see that cytokinesis is beginning to happen, since the cytoplasm is starting to divide to make two new daughter cells. During cytokinesis in animal cells, an indentation forms in the cell membrane along the cell equator. This indentation will deepen until the cell is pinched into two. The cytoplasm and the organelles divide equally between the two halves of the cell. Cytokinesis ends with the separation of two identical daughter cells, which now they can both begin the cell cycle again and enter the G1 phase of interphase. And there you have it. That is the cycle of mitosis. So I hope you all learned something new today. We are going to apply this new knowledge in class tomorrow with a lab. So be sure to check out the Take a Peek section on the website. Remember to rewind this video or look back at anything you didn't understand, and feel free to email me or text me if you have any questions. Take care!